And in this video, we will be talking about gene function, proteins, and enzymes. First is genetic control proteins. First in genetic control proteins is gene-enzyme relationship. Genes encode instructions from making enzymes. A gene's sequence determines an enzyme's structure and function, directly impacting its catalytic role in cellular reaction. The gene-enzyme relationship is a cornerstone of cellular function, operating through the process of gene expression. A gene's DNA sequence is first transcribed into messenger RNA, mRNA, carrying the genetic code to the ribosomes. The ribosomes then translates the mRNA sequence into a specific amino acid chain, guided by codons that specify each amino acid. This chain folds into a unique three-dimensional structure, forming the enzyme. The enzyme's active site, dictated by its structure, allow it to bind to a specific substrate and catalyze biochemical reactions. Therefore, a gene directly dictates the amino acid sequence structure and ultimately the function of an enzyme, highlighting the fundamental link between genetic information and cellular processes. Changes in the gene sequence can directly impact the enzyme structure and function, potentially altering cellular biochemistry. Second is inborn errors of metabolism. A genetic disorders resulting from defects in genes encoding metabolic enzymes. These defects disrupt metabolic pathways, leading to the accumulation of harmful substances or deficiencies in essential molecules. A classic example is alkaptonuria, where a defective enzyme prevents the breakdown of homogentic acid, causing dark urine. Third is the one gene, one enzyme hypothesis. Liddell and Tatum's one gene and enzyme hypothesis, later refined to as one gene, one enzyme polypeptide hypothesis. The one gene, one enzyme polypeptide concept describes the fundamental process of how genetic information directs protein synthesis. A gene's DNA sequence containing the code for a specific polypeptide chain undergoes transcription, creating a messenger RNA mRNA molecule. This mRNA molecule then acts as a template for translation at the ribosome, where transfer RNA or tRNA molecules bring specific amino acids to the ribosomes according to the mRNA sequence codons. The ribosome links this amino acid together to form the polypeptide chain, which folds into a specific three-dimensional structure, forming a functional protein, often an enzyme. Therefore, the gene's sequence directly determines the amino acid sequence and consequently the structure and function of the resulting protein. This process ensures that each gene directs the synthesis of a specific polypeptide, establishing the core principle of the one gene, one enzyme polypeptide hypothesis. The fourth one is protein structure. A protein function depends on its complex 3D structure, built from its primary structure, amino acid sequence, determined by the gene. This sequence folds into a secondary alpha helix and beta sheets, and tertiary 3D arrangement, and sometimes quaternary multiple polypeptide chain structures stabilized by various bonds and interactions. The last is the collinearity of DNA proteins. Describe the direct relationship between a gene's nucleotide sequence and the amino acid sequence of its encoded protein. While largely true for prokaryotes, eukaryote RNA splicing, removal of introns disrupts this direct correspondence. Despite this collinearity undergoes the gene-protein connection.
Good day everyone. I am Alexander Faith Kabakang, BEC1 Agriculture. And today, my assigned topic is all about the protein synthesis central dogma of molecular biology, gene information transfer, interrupted genes. The central dogma of molecular biology is proposed by Francis Crick. Francis Crick was a leading molecular biologist who discovered the structure of DNA and proposed the central dogma of molecular biology explaining the flow of genetic information. The first is central dogma DNA to RNA to protein. The gene for insulin in human DNA is transcribed into mRNA, which is translated into insulin protein that helps regulate blood sugar levels. The second one is the DNA replication, semi-conservative. Before a skin cell divides, its DNA is copied. Each new DNA molecule has one strand from original DNA and one newly made strand. The third one is the transcription, DNA to mRNA. In a liver cell, the DNA sequence coding for the enzyme alcohol dehydrogenase is used to make mRNA that will later help synthesize this enzyme. The fourth one is the mRNA processing 5' polypeptide splicing. After transcription, the mRNA for hemoglobin is modified, adding a 5' and a polypeptide and removing introns before leaving in the nucleus of translation. The fifth one is the translation, mRNA to protein. The mRNA for insulin reaches the ribosome where tRNA brings amino acid to build the insulin protein. The sixth one is the exceptions, reverse transcription. HIV retrovirus use reverse transcriptase to convert its RNA into DNA, which integrates into the host of genome. The last one is the alternative splicing, one gene, multiple protein. The gene for traversing as muscle cells is spliced differently in smooth and skeletal muscles, producing different versions of the trimethylene protein.
My name is Ellen Ansaro Kabakang, base A1A. The topic is gene function, protein, and enzyme. The genetic code, the template code, universality of the genetic code explanation. The genetic code dictates how the sequences of nucleotides in DNA translated into the sequences of amino acids that make up proteins. The process is crucial of life because proteins perform the vast array of functions within cells and organisms. Number 1. The template code. The genetic code is a template code, meaning that sequences of 3 nucleotides specifies single amino acid. Since there are 4 nucleotide bases, adenine, guanine, cytosine, and uracil in RNA, and the 3 position is a codon. There are 43 equals 64 possible codons. However, 3 are only 20 standard amino acid used is protein synthesis. This means the genetic code is a degenerate of the redundant. Multiple codons can code for the same amino acid. For example, both CCU and CCC code for protein. Number 2. Universality of the genetic code. The remarkable features of the genetic code is near universality. The same codons specify the same amino acid as almost organism. From the bacteria to humans, this strongly supports the theory of the common ancestor for all life on Earth. The universality has profound implications for biotechnology because it allows for the transfer of genes between different species. For instance, human genes can be expressed in bacteria to produce human proteins like insulin for medical use. However, there are some exceptions.
Now let's move on to the regulation of genetic code action, regulation of gene action in prokaryotes. Prokaryotic gene regulation primarily occurs at the transcriptional level, controlling which genes are transcribed into mRNA and subsequently translated into proteins. This precise control ensures efficient resources allocation, preventing the synthesis of unnecessary proteins. The primary mechanism here involves operons. An operon is a functional unit of DNA in prokaryotic that contains a cluster of genes under the control of a single promoter. These genes are transcribed together into a single molecule known as polycistronic mRNA, which then be translated into multiple proteins. And ang operon is sa inihiya nga grupohin mga enzyme nga makikita ha DNA han mga prokaryotic cells. Parang usain niya nga switch nga nagko-control han mga proteins nga ang mga gene daman ang mga gene daman ha operon karaan nag-e-encode han mga proteins nga makikita haat metabolic pathways. An operon is consist of promoter, operator, and structural genes. An promoter is the DNA sequence where RNA polymerase binds to initiate transcription. Ang promoter, dito natin makikita ang RNA polymerase. Ang RNA polymerase, usain hiya nga enzyme nga nagtitikang o nag-start ang transcription. Ang transcription naman, ang paghimo hin mRNA tikang ha DNA. Second is operator, a DNA sequence adjacent to the promoter acting as a switch. Regulation proteins bind here to control transcription. An operator naman usahiya nga segment ng DNA nga nag-act as a on and off switch. May daliwat usa nga repressor protein nga pwede dumikit ha operator nga harangan ang RNA polymerase kaya diri makakahimuhin protein. Last is structural genes, the gene encoding proteins with related functions, transcribed as a single mRNA molecule. Ini ang mga gene nga nag-e-encode han mga protein nga related or parehas functions. Kapag na-transcribe or nakopya ang mga ini, makakahimo nahin protein. There are two types of operon, repressible operon and inducible operon. Unrepressible operon, like the trip operon, are normally active but are repressed by a corpuscle, binding to a repressor protein, blocking transcription when the product is abundant. Normally, ang repressible operon naka-on in hiya. Kailangan hin usa nga corpuscle molecule para ma-activate ang repressor protein nga maharang ang transcription pag sobra na ang production. One example of this is trip operon nga nag-e-encode ha mga protein nga naghihim tryptophan, an essential amino acid found in chicken, fish, eggs, cheese, nuts, etc. Kapag damo ang tryptophan, ito ang magsisilbing cortisol. Second type of operon is inducible operon, like the lac operon, and are usually inactive, and lactose inducer activates the repressor, allowing transcription. Efficient transcription also requires low glucose, leading to cyclic adenosine monophosphate catabolite activator protein binding to the promoter, enhancing RNA polymerase activity. Normally, naka-off inihiya kung kailangan hinusang inducer molecule ang lactose para makuha ang repressor protein ha operator ng mahimo ang mga proteins. One example of this is lac operon na nag-i-encode han mga protein nga nagbre-breakdown han lactose. Lactose is a sugar found naturally in milk or milk products. 3 key regulatory molecules that influence operon activity: repressors, activators, inducers. Repressors are proteins that binds to the operator, blocking transcription. Ini ang pumipigil sa paghimo proteins. An activator naman ang proteins that bind to the promoter, enhancing RNA polymerase binding and transcription. Ini naman ang nagpapadili ang paghimo proteins. Last is inducers, small molecules that bind to repressors, altering their shape and preventing operator binding, allowing transcription. Alternatively, they bind the activators to enhance their activity. Ini naman ang nag-aalis han pagpigil han repressor para magkamay-ada transcription. That's all. Thank you for listening and hope you gained something from us.